Okay, imagine this. Trying to send a signal, like a beam of light, straight through a mountain. Seems impossible, right? Most of it gets blocked, scattered, totally lost. Now, picture that mountain is, well, someone's head, trying to shine light all the way through an adult human head, side to side, and actually catch it coming out the other end. For ages, that sounded, well, more like science fiction than actual science.
Absolutely. Just fundamentally difficult.
But today on the deep dive, we're digging into some really groundbreaking new research that basically says, hold on, maybe it is possible. Scientists have actually done something that many thought was pretty much unattainable. They've detected photons, particles of light that traveled clear across an entire adult head.
It's quite the claim.
It really is. So our deep dive today is pulling from a fascinating article about a study from the University of Glasgow. It was published in Neurophotonics. The paper's called Photon Transport Through the Entire Adult Human Head. And it lays out how they pulled it off and what it might mean.
Yeah, the how is definitely key here.
Exactly. Our mission today is to unpack this, understand this like fundamental breakthrough, try to figure out how they managed this incredible feat. And, maybe most importantly, explore why this could potentially open up completely new ways of, looking deep inside our brains, which could impact how we understand and treat things later on.
Makes sense.
Now, you listening are probably familiar with brain imaging, like MRI, right? The big, loud tubes in hospitals.
Standard stuff for deep imaging, yeah.
Well, this research takes a totally different route, using light. And I mean, if this work pans out and inspires new tech, it could maybe make powerful brain imaging much more accessible, maybe even portable down the line.
That's the long-term hope, definitely.
Okay, so let's set the stage. Using light to peek at the brain isn't totally new, right? There's this technique called FNIRs.
That's right, yeah. FNIRS stands for Functional Near-Infrared Spectroscopy. And it's a non-invasive optical method. It uses near-infrared light specifically because that wavelength, it can pass through biological tissue reasonably well without causing harm. Better than visible light, anyway.
Okay, near infrared. And what's it actually measuring? How does it tell us what the brain's up to?
So it works by shining this light into the head and then measuring how much of it gets absorbed as it travels, through the skull, and the outer bits of the brain. It's particularly good at detecting changes in blood oxygen levels, specifically oxygenated and deoxygenated hemoglobin.
Ah, the oxygen carriers in the blood.
Exactly. So when a part of your brain gets more active, it needs more oxygen.
Right.
Blood flow increases there. FNIRS picks up on the changes in light absorption caused by those shifts in oxygenated blood. So it gives us this indirect, non-invasive way to figure out where and sort of when brain activity is happening near the surface.
I see. Okay. So you track the blood flow changes using light, and that maps out brain activity. And the sources we looked at, they highlight some real plus points for FNIRS, the way it is now.
Oh, absolutely. Compared to, say, an MRI machine, FNRS systems can be, well, relatively portable, much less expensive too, which makes them super valuable for certain kinds of research or maybe even some clinical uses where, you can't just wheel in an MRI.
Right. And you can use it while someone's moving around a bit, unlike an MRI where you have to be perfectly still.
Precisely. That's a huge advantage for studying brain activity during, say, natural behaviors.
Sounds really promising, then, for making brain imaging more accessible, more flexible. But there's a catch, isn't there? A big one with current FNIRs.
There is, and it's really been the major roadblock stopping it from being used for a lot more things.
The issue is scattering.
Yeah. As that light goes into the head, it hits all sorts of different stuff, skin, bone, brain tissue, fluids, and it just bounces off in every which way, like a pinball machine.
Yeah.
And the scattering effect is so intense that with the FNIRS systems we have now, the light really only penetrates about, say, 4 centimeters into the actual brain tissue.
Just 4 centimeters?
Yeah, roughly. Before it gets so weak and diffused that the detectors back on the surface just can't get a reliable signal from any deeper.
Okay, let's unpack this. So FNIRS is cool, portable, but hits a wall at about 4 centimeters. That means a huge part of the brain is basically invisible to it, right? The deeper stuff.
Exactly right. And what's fascinating here is, well, why that limitation is so significant. Those deeper brain regions, we're talking structures like the thalamus, the basal ganglia, bits of the hippocampus, the amygdala.
Sort of the core processing areas.
Absolutely critical areas. They're involved in so many vital functions, processing emotions, forming long-term memories, controlling movement, even regulating basic life support. Not being able to see them non-invasively with something portable and relatively cheap, well, that's been a massive barrier. both for fundamental neuroscience research and for diagnosing conditions that start deep in the brain.
So to see those areas now, you basically need the big guns, MRI, maybe CT scans.
Generally, yes.
Hmm.
Expensive, bulky equipment usually found only in major hospitals or research centers.
Okay. So you've got this promising, portable, light-based tech, FNIRS, but it's fundamentally stopped by physics at around 4 centimeters deep, leaving all these crucial brain regions in the dark, so to speak.
Pretty much, yeah.
And that is the wall that this new research from Glasgow seems to have just, well, smashed through.
That's the implication.
Yeah.
I mean, it was genuinely thought to be, if not impossible, then at least incredibly, prohibitively difficult to detect any light signals that had gone all the way through an adult human head.
Right across.
Right across. We're talking light going in one side, being detected on the opposite side.
Right.
Traveling through, what, maybe 15, 20 centimeters of incredibly complex, highly scattering tissue. Skull, brain, everything.
Here's where it gets really interesting then. I mean, how on earth did they even do that? How do you pick up a signal that must be unbelievably faint after going through all that? It sounds like finding a needle in a haystack inside another haystack.
It definitely required some very sophisticated tech and a really meticulous experimental design. The article details it pretty well. The Glasgow team used extremely powerful but also very, very short pulses of this near-infrared laser light. Think like tiny, super intense flashes.
Okay, high power, short bursts.
And then, positioned directly on the opposite side of the volunteer's head, they had these incredibly sensitive detectors, special detectors called SP8s, single photon avalanche diodes. They're designed literally to register even a single photon, a single particle of light that might somehow make it through all that tissue.
Wow. So like the brightest possible, briefest flash aimed at the most sensitive possible detector directly across the head.
That's the core idea, yeah.
Yeah.
And the whole setup had to be in, like, total darkness. Really controlled conditions to block out absolutely any stray light from anywhere else that could mess up the readings.
Right. You're looking for such a tiny signal, any noise would drown it out.
Exactly. They were trying to isolate just those few incredibly rare photons that somehow navigated the entire journey, zigzagging through the whole head.
Okay, that sounds like a massive technical challenge just to build the setup. Did they, like, just turn it on and hope for the best, or was there more prediction involved?
Oh, there was definitely more. A really crucial part of this work, which the source emphasizes, involved running detailed computer simulations.
Ah, modeling.
Yes. They built these complex computational models of how light propagates, how it moves through all the different layers of the head, simulating how billions upon billions of photons would bounce around, get absorbed, scatter through skin, skull, the cerebrospinal fluid, white matter, gray matter, the whole shebang.
So they were trying to predict mathematically what should happen to the light.
Precisely. These simulations weren't just, theoretical guesswork. They were designed to predict what kind, if any, signal they should expect to see on that detector on the far side, if light actually could make it all the way through.
And did the experiment match the prediction?
That's the key finding, yes. The tiny amount of light they did detect in the experiment closely matched what the simulations predicted they should see, which strongly suggested it wasn't just random noise they were picking up. It was the actual, albeit very faint, signal of photons that had completed the full transit.
Okay, that's huge. The simulation confirms the experimental detection, but this raises an important question. Did those simulations tell them anything else? Anything maybe unexpected about how the light managed to get through?
They absolutely did. And this, I think, is where it moves beyond just a technical feat into real scientific insight. The simulations revealed something quite counterintuitive. It turns out the light doesn't just scatter completely randomly and uniformly once it gets inside the head. Instead, it seems to preferentially follow certain paths.
Paths? You mean like specific routes through the brain tissue? That sounds weird. I just picture it bouncing everywhere like crazy.
Well, it does scatter a lot, don't get me wrong.
Uh-huh.
But yes, the models showed these sort of preferential pathways emerging. And these paths seem to be guided by regions within the head that have lower scattering properties, areas where the light is less likely to get bounced off course.
Okay, so some tissues scatter less than others.
Exactly. And the article specifically points to areas filled with cerebrospinal fluid, the fluid that bathes the brain and spinal cord, like in the ventricles or the spaces around the brain. These CSF-filled regions seem to act as sort of, light pipes or low scattering channels that can help guide some photons along, increasing the chances a few might make it all the way across the head.
Wow, so it's not just about raw power and sensitivity. The actual internal structure of the head, these fluid channels, might be actively helping guide the light through. That's fascinating.
It really is, and understanding these preferred paths is super important. It means it's not just a brute force problem. It suggests that maybe future systems could be designed to somehow take advantage of these natural highways for light within the head. It informs the physics of how light interacts with this complex biological tissue.
So what does this all mean then? I mean, this isn't ready for your doctor's office tomorrow, clearly. But it proves a concept, right? It proves something fundamental that could totally change how we think about optical brain imaging.
That's exactly the takeaway. The number one implication is validating the concept. It's the proof of principle. By showing that, yes, light can be detected after going clean through an adult head, the Glasgow team has basically provided the foundational evidence needed to say, okay, designing future optical devices to probe deeper brain regions is potentially feasible.
It breaks through that perceived 4 centimeter barrier, conceptually at least.
Precisely. It suggests that the current limits of FNIRs aren't necessarily the ultimate physical limits for optical methods. So while this study doesn't hand us a new, ready-to-use device, it provides that crucial physical proof. The hope now is that this inspires engineers, physicists, neuroscientists to actually develop new systems that can leverage this ability to detect light through much greater depths of tissue.
Making FNIRS or something like it relevant for those deep brain structures we talked about: memory, emotion, movement control.
That's the ultimate goal, yes. Yeah. To bring the advantages of optical imaging potential portability, lower cost to bear on those deeper regions.
And thinking about the real-world impacts. If we could get a more accessible way to look deep inside the brain, where could that make the biggest difference?
Well, if we connect this to the bigger picture, the potential impact is pretty significant, especially for widening access to advanced diagnostic tools. The source article mentions this could eventually lead to better ways to diagnose and monitor a whole range of brain conditions.
What sorts of conditions are we thinking about where that deeper view using light might be really valuable?
Well, the article flags things like strokes, traumatic brain injuries, maybe certain kinds of brain tumors.
Okay, things that often happen deep inside or affect those deeper structures.
Exactly. And right now, diagnosing those, tracking recovery, often relies heavily on those big, expensive MRI or CT scans. So imagine having a potentially more portable, much lower cost optical system that could still give you useful information from those deep areas. That could be absolutely transformative.
Especially in places without easy access to an MRI machine, like smaller clinics, remote areas, maybe even emergency settings.
Precisely. Think about paramedics or clinics in developing countries. Accessibility is a massive factor in healthcare outcomes globally. If optical methods could start providing some of that deep brain information, that would be a huge win.
Makes total sense. But okay, let's manage expectations here. You said this isn't ready for prime time yet. What are the big hurdles, the current limitations they mention?
Yeah, it's really important to be realistic about where things stand now. This was a proof of concept done under very specific, highly controlled lab conditions. The article points out some key limitations. For one, getting enough signal, enough photons, took about 30 minutes of data collection time on the volunteer.
30 minutes. Wow, okay, that's way too long for any practical clinical scan.
Way too long, yeah. You need something much faster.
Files.
And this is significant. The specific experiment they detailed was done on a subject who had fair skin and, crucially, no hair in the areas where the laser went in and the detector was placed.
Hair and skin pigment would interfere.
Massively. Melanin in skin absorbs near-infrared light, and hair both absorbs and scatters it like crazy. So the source itself calls this an extreme case. They picked conditions that made detection easier.
Yeah.
Trying to get this to work quickly and reliably on people with different skin tones, different hair types and densities, that's a whole other layer of engineering challenge.
Okay, so the big challenges now are speed, getting that detection time down from 30 minutes to maybe seconds or minutes, and robustness, making it work reliably across the whole diverse human population.
Those are definitely two of the major ones, yes. The fundamental physics question, can light get through, seems to be answered with a yes. But now comes the huge engineering lift to turn that yes into a practical, usable technology.
But still, even with those big mountains left to climb, just knowing it's possible, that must change the game for researchers in this field. It tells them it's not a dead end.
Absolutely. It fundamentally shifts the conversation. It moves from, is this even worth trying, to, okay, how do we build it effectively? This research provides that essential benchmark, that foundational data. And those insights about the light paths, for instance, might give clues how to build better systems. The challenges are absolutely significant. The source makes that clear. But the potential reward, especially in terms of making deep brain imaging more accessible globally for diagnosis, for monitoring treatment, maybe even for basic research into how those deep structures work. The payoffs could be immense.
Okay, so let's quickly recap this deep dive then. We started with the current state of optical brain imaging, FNIRS. Good stuff. Portable, relatively affordable, but fundamentally limited to the surface, maybe about 4 centimeters deep.
Right. Leaving those crucial deep brain areas largely inaccessible to optical methods, requiring extensive, bulky machines like MRI instead.
Then came this really groundbreaking work from the University of Glasgow. They actually detected light signals that had successfully traveled all the way through an entire adult head, side to side, something widely thought impossible.
hmm And they did it using a combination of powerful pulsed lasers and these incredibly sensitive single photon detectors, all in a very controlled setup.
And critically, their experimental results lined up really well with sophisticated computer simulations of how light should travel through the head.
Exactly. And those simulations didn't just confirm the detection, they also revealed the surprising finding that light doesn't just scatter randomly. It seems to follow certain preferential paths, possibly guided by low scattering regions like those filled with cerebrospinal fluid.
So the big takeaway isn't a new machine yet, but it's the proof of concept. It shows it is physically possible to get light through the whole head, which suggests future optical devices could be designed to see deeper than current FNIRs allows.
Which opens the door to potentially more affordable, more accessible ways to image deep brain structures for diagnosing and monitoring conditions like stroke, brain injury, maybe tumors, particularly valuable where access to MRI or CT is limited.
But we also have to acknowledge the current hurdles. The demonstration took a long time, about 30 minutes, and was done under ideal conditions, fair skin, no hair. So making it fast and robust for everyone is the next big engineering challenge.
Absolutely. The fundamental physics barrier seems broken, but the practical engineering work is really just beginning. Still, it's a huge step.
It really is fascinating, though, isn't it? Just thinking about pushing back on what seemed like a hard physical limit for using light to see inside us. It could genuinely reshape brain imaging in the future, make it available to more people. And before we wrap up, if you found this deep dive interesting, do consider liking, subscribing, maybe even dropping a comment with topics you'd like us to explore next. It helps us keep these conversations going.
And just thinking about that simulation finding again, those specific light paths, maybe through CSF channels, it really makes you wonder, doesn't it? Could understanding those light highways better not only help us design deeper imaging systems, but maybe tell us something new about the fundamental physics of how light interacts with different brain tissues? Could there be other applications beyond just imaging depth related to these specific pathways?
That's a fantastic thought to end on. What else might these light pathways reveal? Definitely something to mull over. Thanks so much for joining us on this deep dive into shining a light, quite literally, through the complexities of the human head.